Let's continue our discussion of microbial growth and the factors that affect it by considering the chemical factors influencing microbial growth. So, microbial nutrition. We know from our unit on microbial metabolism that organisms, including microbes, use a variety of nutrients to meet their energy needs, things like carbohydrates, proteins, lipids, and even nucleic acids. So they will catabolize those molecules to get energy and also to get monomers, to get raw materials to build organic molecules and then use those to make cellular structures such as cell walls, for instance. And we're going to discuss where microbes get their nutrients from, but we'll learn that it's from a variety of sources, including us if we're dealing with microbial pathogens. To reiterate, microbes use things like carbohydrates and other macromolecules as sources of carbon, as sources of energy, and as sources of electrons. Typically, we take that to mean the hydrogens and their associated electrons. And we, of course, use those to power electron transport chains to make lots and lots of ATP. We learned earlier in our unit on biological macromolecules that the principal elements that microbes need are carbon, hydrogen, oxygen, nitrogen, phosphorus, and sulfur. So those are the biggies, the big six. However, there are other chemical requirements, just like there are for us. For instance, some microbes require trace elements, just like we do, things like zinc. Microbes can also require vitamins, just like we do. For instance, vitamin B12, also known, if you're curious, as cyanocobalamin. Also, microbes, just like we do, require growth factors. Growth factors are going to be molecules, non-vitamin molecules, but molecules that the microbe can't make for itself. Things like heme. And again, when we start talking more about microbial pathogens, when we think about it, it makes sense. That is, why would an organism want to hitch its fate to another organism, like a human, unless that organism has something that it needs that it can't get on its own? So there are, for instance, certain microbial pathogens that have the ability to break open to degrade our red blood cells. Why? Because then they will take the heme, which, if you recall, has that porphyrin ring, and they will use that, for instance, to make cytochromes. So we learned previously that we could categorize, classify organisms based on temperature optima. So for instance, we are mesophiles because we work best at around 37 degrees Celsius. We've also can classify organisms based on their pH optima, and we, for instance, are neutrophiles, being best suited for a pH range of about 6.5 to 7.5. We can also classify organisms based on their carbon source. So, for instance, in our figure, on the left-hand side, we have organisms that can do photosynthesis. That is, their source of carbon is inorganic. They can fix carbon and turn carbon dioxide into glucose, for instance. These organisms are referred to as autotrophs. That is, they are self-feeding. Auto means self, troph means feeding. Other organisms, like us, require organic sources of carbon. We require our carbon to be already fixed. So, for instance, we consume proteins and carbohydrates that the autotrophs have produced for us. We are therefore referred to as heterotrophs. Hetero, other, and troph, feeding. So we would be seen on the right side of our figure, along with organisms such as fungi and certain bacteria. We can further group organisms into categories based on their energy source. So looking at the left-hand side of our figure, we have organisms such as green plants and certain bacteria like cyanobacteria, which get their energy from light. So they are phototrophs, light feeding. Other organisms get their energy from redox reactions involving inorganic and organic chemicals. So they would be chemotrophs and are shown on the right side of the panel. We, humans, would be chemotrophs, requiring our energy to be gotten from organic chemicals. This brings us to the four basic groups of organisms based on their carbon dioxide source and their energy source. So on the left side of our table, we have those that can fix carbon, have carbon dioxide as their carbon source. Those would be auto organisms such as green plants that can fix carbon are referred to as producers. We then have organisms that get their carbon from organic compounds. They are considered hetero, and they are also considered consumers because they must consume, for instance, plants like we do to get the fixed carbon that the plants have produced. All right, so now looking at our top of the table, we have the energy source, and if it's light, then they're considered photo. If the energy source is chemical, then they would be considered chemo. So when we cross carbon and energy source in our first block at the top left, we have the photoautotrophs. That is, organisms that are self-feeding from light energy. We also have, if we look at the top right, We have chemoautotrophs. That is, organisms that can fix carbon, but they get their energy from chemical compounds. Moving on down to our lower left, we have photoheterotrophs. That is, organisms that get their energy from light but get their organic compounds through other means. They get their carbon from organic compounds. Finally, we come to the last block in our lower right-hand corner, and we come to chemoheterotrophs. These are going to be organisms that get their energy from chemical sources as well as their carbon from organic sources. So these would be organisms like us and most other animals, fungi and protozoa, and many bacteria. Some organisms will accomplish this through aerobic respiration, some through anaerobic respiration, and some through fermentation. Now, I know that this is a lot of information, so the organisms that we're going to be most concerned with are going to be the photoautotrophs that we've talked about in lab, organisms like nostoc. And then we'll be interested in the chemoheterotrophs, organisms like us, as well as fungi and protists and bacteria. We have here two critical thinking questions to start off with. The first one is that oxygen is essential for obligate aerobes like us. Why is that? Well, if you don't have an answer immediately, I would refer you to our unit on microbial metabolism, and more specifically to aerobic catabolism of glucose. There are other organisms for which oxygen is deadly. We refer to these as obligate anaerobes. Why is that? Well, it turns out that neither oxygen, O2, nor oxygen that is covalently bound to other atoms in compounds, neither of those is poisonous. However, there are toxic forms of oxygen which are powerful oxidizing agents. That is, they are going to strip electrons, hydrogens, and their associated electrons from other chemicals, and that will then alter those chemicals. So oxidizing agents are going to cause a chain of oxidations of critical compounds like proteins and lipids, and this is going to result in irreparable damage to cells. Think about it. We're always told to eat lots of fruits and green and yellow vegetables. Why? Because those contain antioxidants. What do antioxidants do? They protect us from the consequences of our own metabolism. So let's talk about those toxic forms of oxygen. So if you've been thinking about it, of course, we're obligate aerobes. We absolutely require oxygen because we use oxygen as the terminal electron acceptor in our electron transport chain. So no oxygen, nothing can be done with those protons that we made through running reduced electron carriers like FADH2 and NADH through the electron transport chain. But if there's nothing at the end to accept them, that is, no oxygen, then everything comes to a screeching halt. So in aerobic respiration, in aerobic electron transport chains, when the protons re-enter the cell via the ATP synthase, oxygen is there to accept them. That is, the oxygen gets reduced by accepting the hydrogen ions, the protons. But sometimes during that process, things don't go the way they should, and superoxide radicals are generated. That is, the oxygen is not completely reduced. Now, we aerobes make several enzymes to deal with these oxygen radicals. So we make things like superoxide dismutase to detoxify the superoxide. However, not all organisms get to have superoxide dismutases, and obligate anaerobes lack the superoxide dismutase. And so what happens is that these superoxide radicals, which are all over the place, oxidize their compounds and kill the obligate anaerobes because they can't detoxify this. There are certain mutations in humans that give rise to an inability to detoxify these superoxide radicals, and one of them is a form of ALS, amyotrophic lateral sclerosis, and so there is a familial amyotrophic lateral sclerosis, FALS, that is due to mutant superoxide dismutase. For those who this is sort of ringing up as familiar, amyotrophic lateral sclerosis is also known as Lou Gehrig's disease, after a very famous athlete. It is thought that Stephen Hawking, the famous scientist, may have had a form of ALS, but not the most common form because he was able to live for quite a bit longer than people who have ALS usually do. Now, just because an organism has superoxide dismutase doesn't mean that we're home free, because it turns out that superoxide dismutase, those reactions can create peroxide anion. So peroxide anion can be formed during reactions that are catalyzed by superoxide dismutase. Now, those of us who are aerobes, like us, also have other enzymes to deal with this, either an enzyme called catalase or peroxidase, or both, to detoxify the peroxide anion. So the balanced reaction for catalase is 2H2O2, two molecules of hydrogen peroxide, and then catalase will detoxify the hydrogen peroxide by producing two molecules of water, H2O, plus a molecule of oxygen, O2. The oxygen is going to be a gas. So if we look at our figure, what we see are two slides. And what's been done is to add some drops of hydrogen peroxide to each slide. And then one takes a loopful, so one uses one's inoculating loop, and using good aseptic technique, gathers some microbe and swishes it around in the peroxide. If an organism has catalase, then what we see are bubbles, like we see on the top slide. If we do the same thing with an organism that lacks catalase, then we're not going to see the bubbles. We won't see the oxygen gas, and that is what we see on the bottom slide marked minus. Now, it turns out that organisms that are obligate anaerobes, that is, they can't tolerate so much as a whiff of oxygen, either lack both superoxide dismutase, they also lack catalase or peroxidase, or it may be that they have very inadequate amounts of each. So organisms like Clostridium botulinum, the cause of botulism, are obligate anaerobes because they can't detoxify the toxic form of oxygen. And this is why when we do canning, like home canning, we always do either acid, because that inhibits the Clostridium botulinum. So tomatoes are a big thing to can because they're very acidic. Or we do pressure canning, but where I'm going with this is that it is very, very important that we pressure can low acid foods very carefully, because if we're not careful, then what we're doing is we're creating the perfect conditions to grow Clostridium botulinum. And it used to be very common for people to get botulism from home canned goods such as, potato soup. It wasn't long ago that a number of church members at a church supper got botulism because the potato salad had been made with improperly canned potatoes. In humans, there is a disease called acatalasia, which results from a defect in the enzymes that are required to make the catalase enzyme. And so the catalase is faulty or not made at all, and people who have this have increased periodontal disease, that is, gum disease, and they are also prone to oral gangrene. The causative agent for gangrene is another one of the Clostridium species, Clostridium perfringens. Just like we have classified organisms based on temperature preference, on pH requirements, on where they get their carbon and their energy, We can also classify organisms based on their oxygen requirements. So we can have aerobes. These organisms metabolize aerobically. We can have obligate aerobes that absolutely require oxygen. We, humans, are obligate aerobes. If we look at our figure below, we can use a special kind of culture medium, a medium that contains thioglycolate, which will remove any free oxygen. And so we can look at where in a thioglycolate tube an organism grows. So if we look at panel A, then obligate aerobes grow in the top of the tube where there's lots of oxygen, because if they tried to grow farther down where there's low to no oxygen, well, they would die. They would do the equivalent of us drowning. Now, anaerobes metabolize anaerobically, and obligate anaerobes are actually poisoned by oxygen. So looking again at our figure, we're looking at panel B, and now obligate anaerobes can only grow at the bottom of our thioglycolate tube. Now, there are some organisms, facultative anaerobes, that can metabolize aerobically and anaerobically. And so if we look in panel C of our figure, well, these will grow throughout the tube, but note, they're going to grow much better, more densely at the top. Why? Well, I'm going to tell you another critical thinking thing. Where does one get more ATP? Through aerobic respiration, anaerobic respiration, or fermentation. Finally, we come to panel D, which has aerotolerant anaerobes. Now, these metabolize anaerobically, but they can detoxify the poisonous forms of oxygen. So they can grow throughout the tube, but they're going to grow uniformly because there's no advantage for them in growing where there's oxygen as opposed to growing where there's no oxygen. In addition to requirements for carbon and hydrogen and oxygen and phosphorus and sulfur, organisms require nitrogen. And oftentimes, anabolism will cease, come to a stop, due to a lack of nitrogen. Why? Because to make proteins and to make nucleotides, we need to have nitrogen. Remember, amino acids contain nitrogen, and then our nucleotides have the nitrogen-based ring that forms the information. Now, if one is a bacterium, then one can acquire nitrogen from inorganic nutrients. And looking at our figure, we have a photograph of Rhizobium bacteria growing in the root nodules of a leguminous plant, leguminous peas, beans. And we know from our laboratory unit on prokaryotic ecology that Rhizobium can fix nitrogen. That is, it can take inorganic nitrogen and reduce it to organic nitrogen. So just to jump ahead on our text, rhizobium can reduce nitrogen into and through a series of steps turn it into ammonia or ammonium. This is what we mean by nitrogen fixation. And not all bacteria can do this. It's primarily going to be rhizobium and certain members of the cyanobacteria. And these processes are absolutely vital to life. Now, for the rest of us who can't fix nitrogen, that's strictly a bacterial ability, well, then we have to scavenge for nitrogen. We have to recycle nitrogen from the amino acids in proteins and from the nucleotides in nucleic acids. Besides the requirements for carbon, oxygen, and nitrogen, organisms have requirements for other elements. So for instance, phosphorus is required for making phospholipids, for making the nucleotide bases for DNA and RNA, as well as for the high energy molecule, ATP, and there are some proteins that contain phosphorus. Sulfur is required for the amino acids methionine, met, and cysteine, cis, and these amino acids are involved in the disulfide bonds that are critical to forming the three-dimensional structure of proteins, the tertiary structure of proteins. Also, some vitamins, such as thiamine and biotin, also contain sulfur, and our figure shows the structure of biotin with its sulfur moiety. Biotin is also referred to as vitamin B7 and is a coenzyme for a number of critical enzymes. Also required are some trace elements such as cobalt, copper, iron, and zinc, among others. These we know can be cofactors for enzymes. They are also components, some of them, for the cytochromes that form electron carriers in the electron transport chain. Other things, growth factors, are organic chemicals that various organisms can't make on their own, but need, and these can be various vitamins, certain amino acids. There are a number of amino acids that humans cannot make on their own. Some organisms require the purines and pyrimidines needed for the nucleic acid bases for DNA and RNA, but can't make them. Some organisms require cholesterol, but can't make it. Others require NAD, NADH, can't make it, and or heme that they can't make. Now, when we think about parasites, it makes sense then that organisms that have these deficiencies become parasites. They need it, but they can't make it. So what they do is they parasitize other organisms, including us, as a way of getting what they need.